We'll move on now to the first of our two panel discussions. The panel consists of members of the DTIF consortium, the future of colorectal cancer diagnosis that we've been hearing a bit about from Sergey there. We'll give a few minutes for people to introduce themselves, and if people then in the audience have questions, please leave them below. So on the panel we have Professor Ronan Cahill, surgeon from the Mater Hospital in UCD, Professor Donal O'Shea, head of chemistry, RCSI, Dr. Mairin Rafferty, Chief Operations Officer at Deciphex, and Dr. Pol MacAonghusa, Senior Manager at IBM Research Ireland. So perhaps Ronan, you might kick us off and tell us a little bit about your role in all of this.
Thanks very much, Niall. As surgeons, Niall, we both know that surgery is a really important part of healthcare, and that's Jeff and Savannah were talking about that too. It's still the only cure for many of our commonest cancers. It's a key role in the care of cardiovascular disease, trauma, and so many other conditions. But we know that it's difficult to do and it risks complications. And it's really in all our interest to make it better wherever and whenever it's done. Anyone who's been in an operating theatre, though, knows that we don't generally lack high-end technologies. The key aspect still is the surgical team and the decisions we make. And we need things really that improve our ability to rapidly assimilate all that complex information, personal to the specifics of each patient, with the interoperative findings, and build in our own experience and expertise serially within the time frame of the operation. Our current lack of intraoperative decision support, though, means that even our most advanced current robotic systems have been rather disappointing at achieving better outcomes. And as Silvana was showing, it'd be terrific to bring surgery now into the 21st century by adding to it capabilities that are increasingly standard in other industries. In Ireland, traditionally, surgical research has concentrated on basic science investigations in disease mechanisms, service process and audit type work, but not so much in developing surgery itself, its techniques and its technologies. So at UCD and the Mater, we've been working with like-minded partners to try to develop out this capability, including clinical trials to improve patient care. We'd really love to get Irish patients earlier access to the latest technologies and contribute ourselves to the development of next generation equipment. So one of the things we've been working on for some years is this area of fluorescence guidance surgery. And when I moved back from Dublin, following my work in IRCAD in Oxford, I was so excited to meet Donal O'Shea in RCSI, who's really done a superb work in developing agents that label structures and diseases for surgeons. And together we started applying some dynamic analysis to these fluorescent signals during surgery. And this really revealed some rather remarkable biological insights. But to make it useful, it really had to be teamed with higher end computer vision artificial methods. And Donal and I really had the great fortune to meet with Pol and his team in IBM Research, which kicked off the collaboration that includes the work that Sergey's just presented. Of course, though, the care of patients with cancer includes much more specialisms in a hospital than just surgeons and the operation. So in building out the scope of the project, it made sense to include pathology. And it's been so fantastic too to work with the great team at Deciphex, who've really been doing some cool things in AI pathology analysis. So that's a bit about the genesis of the project, which you'll probably hear more from the other talkers.
Yeah, perfect. Thank you very much. Well, I think we'll go to Pol maybe next. Pol, you could tell us what's IBM doing in all of this.
Well, first of all, it's a privilege to work with lots of smart people. We have a very interesting remit in IBM's research division. As we keep saying to people, we're not a profit and loss part of the organization. Our job is more to apply our research and our technology skills to interesting problems. And when you get a convergence of such, practical and applied domain insight from Ronan and his team, Donal's understanding and knowledge of how dyes work and the work that he's doing on the sort of future generations of dyes. And then, Mairin and Donal, the other Donal O'Shea on the project. We have a lot of Donal O'Sheas. Mairin is, bringing such interesting leading edge AI techniques to image recognition and pathology. It's just a really interesting melting pot to not just try new ideas and develop new technologies, but to kind of reflect back on some of the points our second keynote, Jeff, made. It's to get through that virtuous circle of having people who are really focused on solving a practical problem. So, some people who can bring different skills to the table, and I think that's really important. We're not all doing the same thing, and we're not all trying. I'm definitely not a surgeon, and I think even on his best day, Ronan would probably not try and program an AI algorithm, and nor should he, because we have people there that are very smart at doing it, and then that fail fast mentality. Ronan is a great man for the so what approach to everything we do. We can bring him brightly colored things, but he just goes, so what? And it's just such a fantastic privilege to be able to kind of get that insight really quickly from everybody on the team. So it's a really interesting position to be in, to be able to apply what you're kind of passionate about anyway, because you've spent years training up and building up your skills, but to get that immediate feedback, opportunity to access data, and work on problems that people who really know what they're talking about tell you are relevant and important to solve.
Yeah, thank you very much. We'll come back to you. I think at the end we'll have time for a few more questions on your poll. We won't let you away that easily. Donal, an academic and a chemist, where do you fit in all of this?
Yeah, it's maybe a surprising addition to the team as a chemist, but we actually fit in right at the beginning with the fluorophore itself. I guess what we have to remember is it's the light being emitted by the fluorophore that produces the image that's interpreted by the AI. So getting the fluorophore to do what we want is the key to actually making this a successful project. So what we do is we come up with new and innovative ways to use the existing clinically approved fluorophore ICG, which is part of the work that we're currently ongoing, but also looking beyond that, because we know there are limitations with ICG. We're able to go into the laboratory and make brand new fluorophores from scratch that have specific tailored properties, maybe so we can see specific tissue for its preservation during surgery, such as ureter, or really fancy ones whereby they switch themselves on only when they reach specific tissues, such as cancer. And the goal there is to try to get a good margin delineation around a tumor to guide the surgery in real time in the operating theater, obviously. So it's not just looking at images that were taken maybe hours or days previously. It happens all during the normal surgical workflow to guide the surgical team. But we do recognize and have right from the beginning is that humans interpreting images is very difficult, and the more complex the imagery, the more difficult it is to interpret, and that can lead to then obviously misinterpretation of images. So their AI plays a key role to bring all this very complex imagery together to reproduce it in a format that is actionable by the clinical team. So I guess that gives you a bit of a snapshot of how we contribute across different areas of the DTIF project.
Chemistry doesn't lie.
That's it. No, there's no hiding chemistry.
Absolutely, thank you very much. And finally, Mairin, we've spoken a lot about surgery. But you might explain to us what Deciphex is, which is a slightly different angle, and also then maybe the role of an SME in all of this as well.
Yes, so we're a startup. We set up in 2017. Our focus is on streamlining pathology, so developing a digital pathology platform and embedding, artificial intelligence into that. So, in pathology, at the moment, a lot of it is still done the old way, down the microscope. So what we're trying to do is really make the pathologist more efficient. Our vision is to kind of screen out screen the samples in advance of the pathologist seeing that, and then we prioritize the slides that he sees. So we kind of put the cancers first on the list, and then as they go down by risk analysis, we do that. So this is our kind of a long-term vision. When we met up with the guys in IBM and UCD, they were working on colon. We were like, we're doing exactly the same. So we're developing algorithms for to identify the lesions in pathology in colon, and building out a full kind of platform to streamline the whole process. So in pathology, it has its own problems. So for example, there's a lot more patients with cancer. There's a lot more biomarkers that need to be read. So the pathologist workflow, standard workday, there's a lot more samples, a lot more volume coming their way. So our goal is to really trying to make them much more efficient in their day. So we're not really replacing them. And I heard that the surgeons have the same problem. They think they're going to get replaced. The pathologists have the same worries. So that's not what we're trying to do. We're trying to collate all this information, give them actual quantitative data so that they can make the ultimate decision. So it's kind of a tool, a support tool to help them to kind of get through their workload as well.
And then as an SME in this kind of whole
so as an SME, as I said, we're relatively new. We're only around 4 years. So this was a really good project. The DTIF was kind of national funding for us. You know, as Pol said, that we need the pathologists to help us design this. We are the software engineers. We can build the tech, but we need the pathologists to help us kind of know what we're looking at. Like we don't know looking at a slide what that is. So we needed to be able to interact with the pathologists to get access to samples. So this project was really good in that it gave us access to the pathologists in the Mater. So for us, it's been hugely beneficial for that. As a startup as well, this gave us the ability to do that. It's very difficult to kind of link up with you know, as a small entity. So for us, it's been very advantageous.
Cool. And we have a question here, probably for you, Donal, I would say. How far or close are we from trialing out new upcoming fluorophores in clinical trial setting? For example, perioperative ureteric identification.
So we're getting closer to that position now. So we're just at the latter stage, collecting all the preclinical data to do a dose minimization study. And hopefully we'll have all that data in the next 6 to 8 weeks. And then we have to address the regulatory agencies to ensure that everything is done safely from that point forward. So we're on track for what we said we could achieve in the DTIF, and very hopeful that the first trial in ureter identification will happen in the next 12 months or so.
Great. And maybe a general question for everyone. We're a fair bit into the project now. What's gone well? What hasn't gone well? Or what kind of would be the advice from an experienced panel in this now to anyone else that was thinking of heading along the same route?
I think the spirit of collaboration has gone very well. I think I've been constantly and pleasantly surprised by the openness of people to try ideas. I've been really impressed with the curiosity of everybody to try and see how far we can push the boundary of things. I think some of the things that haven't maybe gone so well, there's just so much to do. and you really do have to sometimes be ruthlessly focused. And there are so many interesting avenues we could go down. So one of the things, that I hope we will see over the next while is the sort of a next step along the road to getting these technologies into people's hands. And, I'll pass it over to Ronan maybe to comment, but, we are looking to scale out lots more data collection, lots more engagement from the surgical community, because we've already started open sourcing some of the tools that we've been developing, and we're encouraging people to sort of come and contribute and engage and be part of the ecosystem. And I would say, we've probably reached a growth point where we've done quite a lot of the preliminary work, in the digital surgery area, at least, and Mairin can comment on the pathology area. But Sergey showed some examples of the kinds of publications we're getting at. We are getting noticed. And in order to get to the kind of next stage, we probably know that we need to expand the ecosystem a little bit from, what we have right now as a base, without necessarily losing the focus that we're able to apply to the particular sets of problems that we're trying to solve. So, I'll pass it over to maybe somebody else to comment on that.
Yeah, no, I think working with people has been the best part of it, and it's really quite inspiring to be pushed to try and do more and do it better. It's been a terrifically synergistic collaboration, and we've needed that because there was a lot of work to do with sort of administrative aspects, contracts and agreements between the different institutions. The university and the hospital, for instance, it's not automatic that an agreement with one happens with the other. So you do really need to draw on a lot of people. And this is really people working with people rather than institutions working with institutions. But you do need to interact well with the GDPR officers at the hospital, the ethics committees. Patients, need and deserve an explanation as to how their data is being used. And yeah, it's been really quite big. Donal mentioned there, investigator-led clinical trials in new drugs, which there isn't a big footprint in Ireland for. But when you have the privilege of funding and such a talented group, there really is quite a lot of pressure then to deliver. You have no excuses. And we're 18 months into the project now, or maybe a little more, and it's great it's on track, but there is more to do. And it's a really, it's a really exciting research project, probably the most exciting one I've ever done, because this is tangible. It's happening already. The clinical aspects are happening already alongside the theoretical and experimental bits of it. So I guess it's de-risked in that sense as it's developing, but also it has all of the real world problems now of doing work in an operating room alongside, the busyness of the clinical environment. And but what's delightful then is to see various people not in the project as such, but clearly also engaged with the project. The theatre nurses are so fantastic, or the porters or the lab techs. It really is. That's your time machine, or your. That's really what motivates you onwards. It's been it's great, but it's also a lot of work.
Absolutely, and you touched on it there. You're a fair bit into the project now. I suppose what can we expect in the next year, or on the home stretch, from anyone really, Mairin, or anyone would like to answer that?
Yeah, from our side, we've built our pathology platform, which is the first step, that will kind of host and manage the slides. The next step is our AI, so we're well advanced with that. We expect that at some point, probably in more like 22, that we will kind of be able to do some validation studies and get those, put them to the FDA for approval. So for us, this is fully aligned with our strategic plan for the company. So we see this as, driving our clinical side of our business. So it's fully aligned. So for us, it's having a huge impact.
Great. Paul, were you going to say something there?
No, I was just going to echo Mairin's comments. We're a research organization within IBM, so our interest is not immediately in commercialization, but more in developing the technologies, learning about the problem, and perhaps generating some IP. And so I would like to see over the next 18 months that we can extend the ecosystem in a number of directions. One I mentioned earlier was building out the sort of engagement for application of the technologies maybe to different problems, collection of more videos, more hospitals, more surgeons. And we're thinking about that very seriously, but also to begin to talk to people that might be interested in consuming some of the results of the work that we're doing, whether that be on the camera side or in other areas related to surgery. Because I think the amount of material that's coming out now is quite impressive. We've made some significant progress now on a good corpus of work that kind of underlines our hypothesis and the science behind this. And I think the task for the remaining 18 months will be to kind of kick on now from, we're much more confident now about the science and the medical application of what we're doing. Now we are going to try and make things stick and see how we can also get them into the hands of real surgeons and begin to have impact on patient outcomes and people's lives.
Yeah, no, that's going to be the fun bit, isn't it? Taking our baby out into the world and seeing how other people react to it. And we have a good advantage of a good clinical network where there's lots of people, Paul, who say, so what, back to you. It's not this important stuff needs to be, treated seriously like this. So yeah, we'll have to build out that clinical network. The initial indications are very good. People are genuinely interested in it. In UCD, we've also built up the engineering side of it to look at optical devices, not necessarily devices that make pictures for people to interpret, but maybe devices that can generate back the signals that go into Sergey's algorithms. And, you know, that's a terribly interesting area, I think, for me, you know, because we're trying to get kind of, you know, useful information to the surgeon, not necessarily just a beautiful picture of it. And surgery is a time of high cognitive burden. So ways and means to support those systems are terribly interesting aspects. I can't wait to see Donal's dies. Hopefully, you know, to see them in a person would just be fantastic, Donal. Hopefully that's next year. And we'd love seeing the pathologists working with the AI and Mairin's. I mean, they've everything from a gorgeous new mouse to this lovely kind of annotation interface. So it's great to get, you know, it's lovely to see the front end. I know the back end is so important, but the front end is what we interact with, and it's great to see that those sorts of things coming forward.
Yeah, and for that, we worked really closely with the pathologists to see exactly what they needed, how would we design it, what works well for them, what does the reporting look like. So all of this came from the pathologists. We wouldn't be able to do it without them. So it was a really good interaction there.
I guess too, we've probably, you know, learned a few things as we've been going through. We've been one of the first projects in the pipeline. And in fact, we were there on that opening day when Leo Varadkar announced the scheme in the Royal College of Surgeons. So not so long ago, but so long ago. And you know, we've been sort of learning how the process works. And as Ronan pointed out, it was quite labor intensive, certainly at the beginning. I think, you know, everybody who's been involved in things like the DTIF has probably learned a few things. And I know that, you know, the processes and so on are a little bit different now. And it has been a learning experience, not just for us as participants, but for Enterprise Ireland as the supervising body and for the department as the, you know, the original funders of the project. So I think, you know, it's been a kind of an interesting, sometimes hard work, sometimes a little bit tedious as we got stuck in processes 18 months to get to this point. But I think a lot has been learned, and I'm kind of looking forward to seeing where we get to in the next sort of 18 months, and then thinking about, you know, what's next after 18 months, because, you know, our DTIF will run its natural course, but there's so much more to do. There just really is so much more to do.
Yeah, so many learnings. But Niall, what's been great too is to see you and Jeffrey and other people coming into the project and making your own kind of advancements in it. You know, that the first year, I guess, was about ramping up what we were already doing, but it's terrific now to see the new offshoots. And yeah, it'd be really exciting to see what other projects can come up. We know we can do these agreements, and we know the institutions can, you know, we can set up the sort of the correct, appropriate pathways to do this type of work, and fantastic for other types of projects to be able to seed or take advantage of that. Now that really could be a terrific next step to the project. Other types of groups and people working together to really try to, you know, to make, you know, the patient care better. And that's really what clinicians are interested in, you know, and that's where we love developing our own specialism into an area by doing new things. It moves us from competency to proficiency, and I mean, it's terrific to be involved in these type of projects. So we'd love other people to be able to springboard from that sort of precedent, I guess, you know.
Yeah, well, I think as a trainee, this is certainly an area that hasn't really traditionally been offered in Irish surgery, something to do with kind of a very clinically based and certainly in the realm of digital surgery. So I think, you know, from speaking to other people out there, that there is an appetite for this, and we can see from today with people that there's certainly the ideas going around. And if we could continue, and that's kind of the theme of the second half of today, is to how do we really keep pushing on from this, and not just this DTIF, but the ones that come on, and anyone else who may want to get involved with the non-traditional kind of science lab-based research areas. So I think that kind of brings us to the end of the first session, everyone. We now have a 30-minute break, kind of stretch the legs and get some food. Reminder, if anyone has questions, please stick them down below and we'll either answer them in the chat or we can answer them live if we get around to it. There will be a recording of this and there'll be material put up online for people afterwards. So we'll see you all back here at 1:30.